So as I mentioned earlier, here for any kind of app, when you go to further information, it tells you what the underlying function is that runs the simulation. Usually it's one. In this case, because we have both the ODE and a discrete time, we have two. So then what you can do is you can basically look up the help file for that function. And on your R console, you can type either help and then the topic, maybe it needs this. I never do it that way. So always do it this way. Oh, okay. Well, so here's a problem. I need to load the package first. Otherwise, it can't find the help file. So it can only find help files for packages you've actually loaded. So, okay, let's try that again. Okay, now it works. So each of these functions, and they all start with simulate. So this way, you know, it's one of these underlying functions that are running. They're all starting with simulate. This basically gives you the explanation of, you know, a short description, what's going on. It's not as detailed as you see in the graphical interface. So really, you probably want to read, you know, all of this part here first to really understand what the model is doing. But once you have that, then this gives you a quick explanation of how it works. It tells you here, and that's, you know, if you're new to R, this sounds maybe very confusing, but you'll get the hang of it. So this is how you call it, and this is the things you can send into the function. And if there's something behind, that means it actually has a default value. So if you call that function without specifying a value for, say, B, then it will just use the number 100. A lot of times they all have default values, which means you could call this one without specifying any of these and it would work okay. Okay, increase text size. I can do that. Let me see. Okay, you're one step bigger. Let me know if that's still not big enough. And here's a quick explanation. Basically, these things that are sent into the function are called arguments in R. So it explains, you know, what they are. This is B, and that's the starting value of a bacteria, and I, and so on. So it tells you what it is. And here's a little bit more details. And then it tells you what you get back when you call that function. And pretty much always you get something back that's called a list. If you're new to R, that might not tell you much, but it's basically a data structure that is fairly flexible and stores the information you want. In this case, it just stores basically the time series, basically, oops. It stores the numerical values here for B and I and the time as well. That's what you get back from that function in this case. And really good, anytime you start a new R function, I almost always go right to the bottom and look at the examples. The examples are really good at showing you how you can call that function and how to do it. So in this case, here's an example. You can call this like so, and it should run if you have the package loaded. So you need to load it first. Now I can keep typing stuff here into the console, but that's actually not, that's kind of bad coding style, because then I lose it all. So what I will do instead, and again, you can all work along with me if you want to do this on your own. I will start just a new R script, which is basically just a file that contains all the commands. And the good thing is then I have it and I can save it and I can open it and go back to it. And I also can run all of them at the same time instead of having to type one line in here. So I go to new file and I'm just going to use a basic R script. So this is here, it. And now I copy just this command over there. So first on my script, I load the package. And it's very good practice to always comment your code. I always tell my students, you need to have at least 50% of your code needs to be comments. So you will thank yourself 3 months from now when you're wondering, what in the world did I do there? And also if you work with other people and share code, it's really important to have that commented. Otherwise it can get very confusing. So I typed them in. Now what you can do here, I might need to give this a name. Just call it R script. And then what you can do once you have a long list of commands, you can hit the source button that runs all of them, or you can hit the run button, which kind of runs one line. You see it's run down there. Now, one thing that's nice when you use RStudio is that it shows you over there in the environment what you're getting back, so you can actually click on it and look into it. There's an equivalent command, or there are multiple commands. One that's built into R is the str command for structure. And when you type this, you see it tells you here a little bit what's inside. Now, that might be a little bit confusing, but the point is, the important one is here, there's a data frame, and what that means is basically just a matrix. You have columns and rows. In this case, you have time, the number of bacteria, and the immune response. And so those are, we could print them now on the screen, but, you see it started printing it, but it's already too much to look at it, right? But you get the idea. We can just look at the first few. There's a command called head in R. So you see it's time, bacteria, immune response, and then that's how they change in time. And a lot of the apps have that. You get the time series on. And that's the fundamental thing when you run these kind of models. That's what you're getting out of them. The change over time for the variables you're tracking in your model. Everything else you compute from them, like we saw earlier, the maximum or the steady state or anything is done from this sort of raw material that you get out of your models. So I'm just copying this over there. Now what we can do with it, we can plot it. The graphical interface, the package has some helper functions that let you plot it. For that, you have to get it in a certain structure. I'll share that with you, but it's good to just be able to do it yourself. And for that, you can use base commands for plotting. So just the plot command in R. Actually, let's make it simple. So it returns it as a list, this thing result, and we only want the data frame that has the time series in it. So I'm just doing it for simplicity. I'm just pulling it out of there. So I have to type less down here. I could always type it full like that. In fact, let me just do one line with it and then without. So I'm now putting time on the x-axis, and I'm going to put the bacteria on the y-axis. And this just L tells it to plot a line. So this is what I get here. It doesn't look as pretty as we saw through the graphical interface, but it's basically it. It's the bacteria and their change over time. And now I can, if I want to, put the bacteria on there. So now I'm showing you, I pulled the TS out. So now I just have to type less. But you can always type the full thing if you want to. And so I can distinguish it. I just give it some color, different. Okay, here now you see the immune response below it. So it looks like the plot you saw through the graphical interface, but now you're doing it completely yourself. The nice thing is, of course, now you can do all kinds of stuff to it. So, if you wanted to turn that into a publication figure and you needed it to look a very specific way through the graphical interface, you can't do it, but here you can. There's also another way. So the package has the function called generate ggplot that basically makes it. It has a very specific way in which you have to feed the result into it. So it has to be sort of a list of lists. That's just a way so it can process all apps. So, if you want to know more about that, you can look at the help file for this function. And so this is now a helper function. So it's, you might not really need it, but sometimes you might want to do it. And it's a bit complex. So it has this structure and it has these very specific settings. So maybe not, that interesting initially or useful, but I'll just show you. So here you see this now looks basically exactly like this because that's what we did. We just now did it ourselves with that function. So you can call that.
Hi, excuse me, Andreas.
Yep.
Before you've gone any further, so once you load the library, and so did you pull some data into R? Like how
No.
you got these results?
Oh, I ran my simulation here with this code here.
Okay.
So we can actually, let's do it more explicitly so you can see how it looks.
I did that, it didn't work for me. So maybe it didn't work for me. I tried to run simulation just like yours.
This and this, and it didn't work?
No, I think I might have missed something. I can
should work if you call these codes. But let's make it more explicit here. So as I said, there's a lot of defaults that are just set. That's sometimes a nice shortcut. But often it's also good to specifically set so now we know really what we call the model. So this tells you explicitly we called the model with this many bacteria and this much immune response at the start, this growth rate, this carrying capacity, this decay rate, and all of those. And so this is the default setting. So this will now give us exactly the same figure. So you see it was the default setting as above. But we can now change around. And so we can now, say we want the growth rate, that's what we did earlier, to be faster. So you just call it with another value, you run it, and you see it looks different now. So that's what we earlier did through the graphical interface. We over here, we set G to 2 and hit run. And basically, that's exactly the same we just did, just now with code. We typed this in now and got this. Also earlier, you saw in this one here, oops. Something's weird here. Okay. You saw down here, it gave us the minimum, maximum, and final value. So we can do that ourselves. Basically, what we need to do is we need to take this big TS object here, which has all the numerical values for B and I, and pull those out ourselves. So, we could do the maximum. There's a function called max in R, not surprisingly. So that's the maximum of B. If we run the default again, we can compare with what we had earlier. Oh, it is already the default, so let's compare. Yeah, same number here. You see, that's basically what it does under the hood. It just uses the max function to pull that out. And then for the final state, it just finds the one at the end. So you saw earlier there was this command head that looked at the beginning. So you wouldn't be surprised to learn that there's a command called tail that looks at the last 5 entries. And so, we're just pulling out B at the end and I at the end. And those are our values at the end of the simulation. They might or might not be a steady state yet, but those are the ones at the end. So that's how you can, interact with the simulation functions yourself. The disadvantages, we've wrote a few lines of code, so we're actually now coding. The advantages, we can do more stuff now more easily than we were able to do before. Because you have now the whole time series, right here, all it returns to you is the minimum, the maximum, and the final value. But let's say what you really want to know is you want to have the time of the first maximum. Like getting it out of this thing is just not very convenient. But once you do code, you could actually do that. Now for that, you need to start looking around a bit in R and figuring out how it works. But so R has a which.max that basically tells you at what entry, in this case, in which row does the max happen. So it tells me in row to 1099. And now what I can do is I can now figure out what the time is at this index. So I go into that row and I want the time now. So I'm pulling out the time. So if I copy this, okay, I need to run it first. So I ran this one now. So it tells me after 21.98 days, that's where the maximum occurred. So you see, I have much more control now if I use code. I can get stuff that the graphical interface doesn't give me easily. Seems about right somewhere here is the max. Of course, I could do that for the same for B, for I. I would just call, this would be the max for the I variable. And that happens apparently at 24 days, a little bit later. So I think you get the idea here that you can run the underlying simulation, and with not too hard, too difficult code, you can pull out more than you could with the graphical interface. And that's the idea here. You can get one step further. You can explore somewhat more. Now the drawback is it's still a model with bacteria and immune response, and that's it. You can't change it. Well, we'll get there, but you can't change it currently. So, you can explore this underlying model, but you can't at this stage modify it. Let me stop here for a minute and see what kind of questions or comments people might have. So if you've, no, yep.
Sanjay, when you copied this code from Shiny app, what tab were you in? Sorry, I missed that.
Say that again.
Like you copied these codes from the Shiny app, right?
No, I wrote this code.
Or yeah, the first one, I thought like row 20, like B and B max. I thought you took it from.
Oh, this one here?
Yeah.
Well, I quickly cheated and copied it from the solutions. It's not the Shiny app, and I can share that with you too. In fact, okay. Let me show you through that real quick. Not quite all, we're working on it. By the end of the course, hopefully all the apps will have full solutions. For obvious reasons, they're not public because, if I want to teach with it and have students submit it, I can't have them public, but I'll definitely share this one with you and maybe share others that we go through. Basically, in the solutions, I'll do it exactly the form that I just showed you instead of, I can't really show solutions with the graphical interface unless I record myself doing it. And that's kind of, not that ideal. So I'm doing it here, actually just calling the code. So what I ask, you to do here, the what to do, which most of you will probably be doing graphically. If you go through it, you could also do that completely in code. So you could try to do all these tasks without touching the graphical interface and just writing code. And this is what I'm doing here. So, here in the first one, I just called the simulation. Then this, I just explain a little bit what I just told you, how you can get different things out of there. And then, here I show how you can get the final one out of that. And that is what I've asked, people to report. So that would be the answer. And so the same for the others here. I'll show how to do it with code on a log scale. Here, Plotly one. So basically, this is just code. If you copy and paste these pieces into your R console, you can reproduce it. And there's no Shiny involved here anymore. So Shiny, meaning that graphical interface part here, is completely gone. It's still the package, and it uses these functions from the package, namely simulate ODE and also the discrete one. But you can do it fully in code. So you could try to go through the first app here and do all of these tasks by just writing lines of code. So that's what I did. I was just lazy and copied it from the solution, but I could have also just looked at the help file and learned here that this is what I need to put in. So I could just copy this over there and call it with those settings or whatever settings I want. So usually you pull up the help file, it tells you how you need to call it. And then, you know, we can do that now for the discrete one. So you think it's basically the same because it's not that much different. It has the same settings that you call it with. And so I could copy that now over from the help file. Could put it in here. I'm not sure if the formatting works here, but let's see if it runs it. So this now ran the discrete model. If you look at it has the same structure because it runs the same model. So again, they're just bacteria and the immune response and time. So the underlying model is exactly the same. We're not going through it here, but you know, if you go to the next app here, there's a basic virus model, and it's conceptually similar. Only instead of bacterial immune response, it tracks uninfected cells, infected cells, virus. So if we're interested in this, we could figure out that the underlying one is called Simulate Basic Virus ODE. And so then we could look at its help file and we learn that this is how you call it. Again, there are default values for everything. So I could actually just call it without setting anything, but let me just copy it to be completely explicit. Copy it over there. By the way, I'm just lazy and call that result here every time, but you can call it whatever you want. In fact, if you want to keep track of it, you might want to call it something else. Let me just, for the heck of it, now call it virus result. So we know this comes from a different model. So I run that. Now, if I look at this structure, you see it's a little bit different because now I have 3 variables that I track. So now I get time again, and then I have now uninfected cells, infected cells, and virus. So every time you get the time series for all the variables you have in your model. That's always sort of the raw material that it returns to you. And of course, you could now do the same kind of plot approach here. And we did. I'm going to be lazy and call it TS again. This is just pulling out the data frame that I need. Really here, it's just kind of sticking the data frame inside a list is not actually that useful in this case. It's just for generality. So my generate functions underneath do everything. That's why this thing has added complexity that's in some sense unnecessary, but I need it. So now I want uninfected cells, infected cells, and virus. Plot those 3. Okay, those are those. You see, there is a problem with just the basic plot because it actually sets the scale based on the first one here. And then the other ones are not looking so good. So plus we might probably want to plot it on a log scale. So you can do all these changes. In general, I recommend that, you know, when you make plots, especially if you're thinking about plots for research purpose and for papers, the basic plotting function it's harder to get really good looking plots. You can do it, but it takes much more fiddling. So I would generally recommend use ggplot then, the plotting commands. So it takes a little bit to get used to it. So ggplot is basically that package that makes these kind of plots here. Plotly is not so good because Plotly is just interactive, which is nice for web pages. But if you want to have a figure that you can put into a PDF or PNG to give to a journal, then you want some different output, and interactivity doesn't work anyway. So then you might, ggplot is really a very good option. And there's so much help on ggplot out there that, you could really, without much problem, probably was just a little bit of Googling, figuring out what to do. Try to do that. See what that does. Okay, that was too much. Shouldn't have quite these extreme. Maybe that. Okay, looking a bit better. So that's for a virus model. So the thing is always the same. You run the underlying simulate function, and then you get something out. So there's a question. So there's a question that's a bit technical, but the answer is yes. It's some kind of ODE, something like Runge-Kutta, but it's not Runge-Kutta. It's an implicit solver, because sometimes the equations get stiff and then the explicit doesn't work so well, but maybe that's too much technical detail. So it runs some kind of underlying ODE solver, and discrete time model, it just steps through in time. In fact, we could look at that. Well, we'll get to it tomorrow, but we can look at it real quick anyway. So if we look at the discrete time model here, you see there's just a loop, a simple for loop that steps through time and does it, versus the other one we're calling another package in R called DESolve, or the package is called ODE that runs the function is ODE, the package is DESolve that runs the underlying function. We'll look into those in detail more tomorrow. Right now, we're kind of treating those things here, these simulate functions, as black boxes. Another question, how do you know that the initial values of the parameters are the true values? What do you mean by true values? They're just numbers I made up and put in there.
Well, I meant when you don't know the initial value of your model, how do you know that this value is actually correct for your data? It's actually correct for your model to describe your data?
Well, that's kind of a scientific choice here. So that actually where you will spend a lot of time once you build your models. You have to go out and find good values for those parameters based on known biology. And that's where these models often stop working. If you don't know enough about your system that you can't find good choices here, then maybe it's not ready yet for using those kind of models. So, you will, usually the starting values are not that important, but, let's go back to our bacteria model here. You assume there's a certain amount of bacteria that start an infection and there's immune response at some level. Then based on biology, you know that the bacteria, double every 12 hours or so. That can give you an estimate for the growth rate. You know that they usually kind of level off at something that people have measured. So that's your carrying capacity. But basically, you need to look into the literature or do your own experiments if you're an experimentalist and find values that are reasonable for those. And you'd be surprised. Most students are surprised when they start modeling how hard it is to actually get hard estimates for a lot of these things. Things that are actually not that hard to measure, but no one has ever bothered to actually measure them. And sometimes the best you can do is you say, okay, I want to model the growth of strep, but no one has measured strep and staph is similar. So I'm just going to take the growth rate that I found in a paper for staph. Or E. coli or something like that. So that's where they come from. So basically it comes from scientific knowledge that you have to find somewhere. If you're lucky and you find very precise ones, you can stick that in. Sometimes you can only find ranges, and that's something we'll get to tomorrow. Then you can do this thing here, uncertainty and sensitivity analysis. Basically what you're doing is you're changing your parameters over ranges and see how that affects the outcome. You're saying, I really don't know if the growth rate is 1 or 2 or 3. So let me, sample over range and see what the outcome is. Or you can do the systematic exploration that we saw earlier, where you basically change G systematically from one to the other while keeping everything else fixed and see how it changes. But yes, coming up with good values for parameters is tricky in this example because these are just toy models and they're not trying to be any exact specific disease. It doesn't matter much. I just picked some that gives overall okay patterns that kind of look nice. But yeah, when you want to try to say, okay, I'm actually now modeling pathogen X or pathogen Y, then you need to try to, carefully match those. So this is if you don't have data. If you do have data, then you can use those models and try to estimate some of the parameters. You will still only be able to estimate a limited number. So for this basic model here, if we only had measurements of bacteria over time and try to fit, we could probably at most fit maybe 3 of those parameters. But you see, we have 6 here. So we might still end up having to fix some of them. So most of the time you'll have to pick some that you fix, and you have to do that based on your best understanding of the system. That again is where modeling is not a recipe, but is a bit of an art there that you decide what's reasonable.
Okay, thank you.
Okay, there's another question. Once you put some parameter values and run in Shiny app, is it possible to copy R code that ran behind the scenes? Hopefully in the future. It's high on my list to do that, to have a button where you run your simulation and then there's a button here that is download code, and that gives you the actual code that reproduces this. And there's a prototype, but it's not fully working yet. So not yet. Whenever I get around to finishing and implementing that, then it might be possible. So currently not. But it's definitely would be something useful because you really want to be sort of reproducible. And that's the problem with the graphical interface. You can't really be very reproducible because you don't remember what you clicked. That's why I think it's okay for teaching, but I think for research, you need reproducibility. And therefore, then this way of doing it is much better, because if you open that same script again several weeks from now and run it, you should get exactly the same results.
Can I ask a follow-up question on that?
Yeah.
So will you recommend for having codes for future, then pull out R and then try the same parameters here and just compare if the output is the same like in Shiny app, because you would like to keep codes for future for research or publication?
Yeah, I'd recommend if you start using this for research and publication, you'd probably want to stay away from the graphical interface completely, and just do it this way with code, and not even go to the graphical interface anymore. So I think this is mostly for learning and getting the hang of it. But once you understood that you can do all of these things here in these tasks by writing code like this here, then you probably just want to do it this way. I think that's the better way then. And also it makes it more flexible and powerful, and you likely will soon hit the limits of the graphical interface. If you want to do something that the graphical interface doesn't let you, then you're just stuck. But in this way, you might be more likely to be able to do things that otherwise you couldn't do.
Great, that's helpful.
Other questions? You can unmute yourself or put it in the chat. Let me go there real quick, and then I think we'll revisit that tomorrow. So that's the other one we explored earlier, where it's samples over parameters. And so we again looked at what the function is called, and then I can look at the help file. And here it tells you again the sample and what you put in. You see you put in some more stuff here because we have to tell it how many samples, where we start, which sample parameter we want to sample. If we want to sample it linearly or log space, there was a question about that earlier. So you can set that here, how you want to do that. So if we ran this, copying over here, takes a bit longer because I now run it 10 times. Now, if you now look at, you see it looks different. It's not the time series anymore. And the reason for that is that this function itself calls the underlying simulation model that we just looked at, which runs the time series. But then once it has run the time series, it just extracts the peak and the steady state for these different values. So you see now here that data frame is actually different. It has the X values, which is the parameter here we want to change. And then for each one of those, it reports the peak and the steady state. So now if you plotted a plot here, it would look similar to the ones we had earlier. So we're, you see it's now not called TS anymore because I use TS for time series and it's not a time series anymore. So I'm just calling it DAT for data more generally. Though it's simulated data, of course, not experimental data. So now I'm plotting the X value. In this case, it's always called X value. So I need to look at the function call to actually know which one I did. So in this case, that was, I sampled over parameter G. And let's say I want to see how B peak changed with it. For here, points is maybe better. And I don't think I need those. So let's just look at that. So that could look kind of similar to what we've seen before. Of course, not quite as pretty, but you know, you can see sort of the overall idea here. We're looking here at, I think, B peak here. So that's what we're here. And of course, it could plot the other four now: I peak, B steady, I steady. And then it also returns that steady one. That is basically this one here that tells you at the bottom system has not reached steady state. So it's basically just a warning telling you, hey, you didn't run the simulation long enough to have actually reached the steady state, which means you should take these B steady and I steady values with a grain of salt. So different functions will return you different things. For several, it's time series. For some like this, and then the fitting ones, the structure that's returned is a bit more complex. But always when you look at the help file, it will tell you under value what is returned. And then you can sort of always look at it and hopefully from that know what to do with it, how to further process. Of course, for that, you need to start getting used a bit to R and its data structures like data frames and lists and things like that. So that requires a bit of learning R code. But it's still fairly basic code.